Great.
Good.
Let me see if it's sending me. It's saying it's live. Yeah. Oh, it's a new link though. I think. Sorry. Do you find the link?
I'm just seeing if it goes to it.
Well, it's recording and then it looks like we can
Okay, I'm gonna play with it and then you can hit start. I'll just flip it around. What I don't know is
if you go to my YouTube account, just go to like
I think okay, I'm logged in as you.
yeah, and then go to live videos. No, I think go to Creator Studio. Yeah, right. Yeah, and
This is where
is that the new one?
we have a new one. Great.
View all 6. You know what we should do? We should just create a brand new and then like just send out that link. So end the stream. Oh, there you go.
I'm such a
Never mind.
dingus. Sorry about that. Copy. Put it in. Sorry. Okay.
Thank you for your help.
Okay. Oh, you're live streaming on YouTube?
Oh, hell yeah. Well, yeah. Via Facebook.
Great.
I think we'll get started. There may be some other people coming in, or they'll have trouble finding a seat. So it is one of the great thrills as a PR to celebrate these moments with our students. And it's really an absolute thrill to be here today to celebrate Scott's achievement in graduate school. It has been tremendously fun to have Scott in the lab. When he, as those of you know him, can already imagine why. When he came into the lab, he took on an ambitious project, working with Aaron, postdoctoral fellow. The two of them, dug into something that in retrospect, I don't think we didn't quite know what we were getting into. But Scott just did a tremendous job, worked super hard, was really kind of outside of my area of expertise, found the help he needed, and ended up, really sort of succeeding in a new area for us. As often happens with graduate school, there was that kind of, valley of death period in the middle where, things looked a little dark. But Scott soldiered through. I always had real confidence that he would, and he did. And it was just super exciting. It made it all the more satisfying when in the end, he really put together a beautiful story, and you're going to get to hear that today. And I'm just super proud of the work Scott did, the effort Scott put in, and I'm really glad that you're all here to see that. Personally, having Scott in the lab is just great. As those of you who know him know, he has this wonderful, quirky sense of humor that it's definitely going to miss. Absolutely. The bonus of having Anshu as a kind of honorary member of the lab and your delicious Hamantaschen that I hope you'll send us some. And then even the cat videos I'll miss just a little bit. Just a little bit. For those of you who don't know, Scott got a great job. I'm sure family's excited about that. And I'm super proud of him for that too. So it's just been really fun. I'm going to miss you, Scott, but you're ready to go. So let the people see it. speaks to Aaron's intelligence that we get the crying out of the way in the beginning, which I was not expected for, but maybe it's better in the long run. So now I'm going to struggle through this whole thing. Thank you. Okay, so yeah, as you know, this has kind of been a long time coming. A little too long for some impatient people. So we will get right into it. So our lab studies neurons, and neurons are the functional component of the nervous system. And I have this cute cartoon of what a neuron would look like if it was in a cartoon show. It receives messages through these slender processes called dendrites. It processes them in a cell body and then transmits that message via an electrical signal down a long, slender process called an axon. And when that signal gets to the end and reaches a synapse, or the connection of that neuron to its target, which is usually another neuron or a muscle, then that next neuron or muscle will either contract or pass on that message. And our goal in the lab is to really understand the biology of the axon and how it responds to injury and disease. Axons bundle together to form what you know of as nerves. And here I have schematized the human nervous system with the brain and the spinal cord shown in red. That makes the central nervous system. And all of this blue is the peripheral nervous system. And you can immediately appreciate how long some of these blue lines are. These are peripheral nerves. And indeed, you have some neurons in your body that have cell bodies that lay right outside your spinal cord in your back and extend axons all the way to your extremities. So right now you have some cells in you that are over 3 feet long. And to better illustrate that, instead of this cartoon, this is really what a typical neuron can look like, where you have the cell body, and to scale, the axon is an extremely long, slender process that can reach enormous distances. So to put this into perspective, if we took this cell body and we blew it up from its normal microscopic size into the size of this room, the axon would extend roughly to Chicago, which is about 300 miles away. And so I hope this lends an appreciation to how long axons can possibly get, which on one hand is really amazing. And that's pretty awesome that a single cell can extend that amount of distance. But it also makes the neuron vulnerable to damage, because if you have this enormous structure, it's inevitable that it can be injured in a number of ways. And to make a long story short, what happens if an axon is injured? Well, it's not good. The axon can't function, or the neuron cannot function without the axon. So if you think of an electrical piece of equipment that has wires in it, when you start snipping the wires, it's no longer going to work, especially if one of those wires controls something very important, like breathing or heart rate. And so axon injury can occur from a variety of sources, including physical trauma, such as accidents or traumatic brain injury, but also a variety of neurodegenerative diseases, such as ALS or Alzheimer's. And axon injury is a particularly challenging thing to deal with because of two main features. One of them is that neurons are postmitotic, and this means that they don't divide. And so normally when other tissues in your body are injured, such as your skin, you get a cut, you have skin cells that are around the injury, they divide to make more of themselves, you get new skin, and now you're repaired. But you can't make any new neurons, so they can't do that. For the most part, the neurons you're born with are the neurons you have for the rest of your life, which on one hand is really amazing, because that means there's some neurons out there that are over 100 years old, still working the same way as they did when they were born. However, because you can't make new neurons, existing neurons are forced to deal with axon damage and thus repair the axon to restore nervous system function. And the second complication is that central nervous system neurons in the brain and the spinal cord can't regenerate their axons. So once they're injured, the neuron is shot. And that's why things like spinal cord injury are so devastating, because once those axons are gone, whatever function they promoted, like movement or breathing, you don't have that function anymore. And so, fortunately, all of this blue, the peripheral nervous system, those neurons can regenerate their axons. And so we use that as a model to understand how neurons have the ability to regenerate axons in hopes that we can understand all of the molecular mechanisms or all of the microscopic things that are happening inside the neuron to enable repair. And then once we understand that, perhaps we can develop therapies that promote regeneration in things like the central nervous system where that normally doesn't happen. So I'm going to quickly walk you through using this very elaborate cartoon of how a peripheral axon will regenerate. So here we have the cell body, which contains the nucleus, and here's our axon. And once the axon is injured, a number of things are going to happen. First is the degradation and clearance of the distal stump, or the part that's no longer attached to the cell body. Next, a number of things are going to happen at the injury site. One is the formation of a structure called a growth cone. And essentially, this is a finger-like structure that enables the axon to pathfind and grow. At the same time, a number of proteins localized in the axons are turned on by this injury. And we call these guys injury signals. These injury signals are then going to be transported back to the cell body. And while they're on their way, it takes some time to get there, there's a little bit of regeneration that occurs out at the injury site. However, importantly, when these injury signals get to the cell body, they converge and drive a massive upregulation of genes, or basically they turn on a bunch of genes that promote axon regeneration. And collectively, we call this group of genes the regeneration program. And once those genes are on, they pump out all the raw materials required for this axon to robustly build and extend new axon. And then this is where you get the robust axon regeneration that occurs in the peripheral nervous system. And so the goal of our laboratory and many others is to identify what these injury signals are. So how does a neuron know it's injured using these signals? And how do they drive this program that enables robust axon regeneration? Because if we can figure that out, how these guys work, then maybe we can turn them on using a therapeutic approach to promote this robust axon regeneration. So how do we identify injury signals in an experimental context? Well, like most things in biology, we break it and see what happens. And so in the context of injury signaling, what we would do is we remove an injury signal and then ask, can we turn on the regeneration program? And if we can't, then by deduction, whatever we removed is an injury signal. And then we can further study how that thing works in this process. And so we can assess whether the regeneration program is on in two main ways. One, as I said, the program is a massive collection of genes, and we can probe for those genes and ask, are they on? Alternatively, we can look at the functional readout. And the function of this program is ultimately growing more axons. So we can look out here and ask, are the axons growing? And so I'm going to walk you through a really cool paradigm that we use, that we can specifically use growth as a readout to identify new axon injury signals. And that's called the preconditioning phenomenon. And I'll walk you through it here. So here's our neuron with the axon. It's going to be injured. Injury signals are activated, sent back to the cell body. Once they get there, they're going to drive the regeneration program. Well, how do we really know that the program is on? Well, one thing we can do is injure the axon a second time. And now, because the program is already on, it doesn't have to go through all of this business again. It's ready to go. And indeed, we see immediate, robust axon regeneration. And so this is a really cool trick that enables us to assess whether the regeneration program is active. And so how do we use this in practice? Well, I'm going to walk you through some data that we collected previously in the lab to demonstrate that this kinase, called dual leucine zipper kinase, or DLK as an abbreviation, is one of those axon injury signals. And DLK is highly expressed
in the nervous system, in a variety of organisms, and it's absolutely essential for this process. Well, how do we know that? Well, we can use our cool preconditioning trick. So here's what it looks like in a real mouse. So here, what you're looking at is a nerve, which again is a bundle of axons. So each of these white lines is a single axon in the nerve of this mouse. This yellow line is the injury site. And you can see if we crush the nerve here and look 24 hours later, there's a little bit of regeneration. These axons have moved past that injury site. However, what if we precondition these nerves? Here, we injure the nerve, we wait a few days for the regeneration program to turn on, and then we give a second injury and give the same 1 day worth of time to allow the axons to regenerate. And now they look like this. So these axons have been growing for the same amount of time, except in this nerve, the regeneration program was already on. So now what happens if we do this in a mouse that does not have DLK in these neurons? Well, now it looks like this. So you can see there's a little bit of regeneration past the injury site, but now you don't see this preconditioning effect. So essentially what we've done, what this data demonstrates, is that DLK is dispensable for this limited early phase axon regeneration, but is required for this robust late phase of regeneration that depends on this axon regeneration program. And thus we can conclude that DLK is one of these injury signals. And so we wanted to find new things that work alongside DLK or work like DLK, because most times in biology, it's not just one gene or protein that's giving you a phenotype or a result. It's usually a collection of things working together. So we wanted to find more. However, that in vivo assay that I just showed you is a really great tool for looking at one gene at a time. But we wanted to look at a lot of genes at a time. And if you want to look at a lot of things, you can't really use mouse models because they're expensive and they take a lot of time to make. And so we wanted to find a way, can we look at a whole bunch of things at once and find a collection of new axon injury signals? And now we're in the dream scenario of every geneticist because we get to do a screen. And so that's where my project comes in, is can we take that in vivo preconditioning assay or that double injury assay, turn it into or transmit it into a dish, and then look at a whole bunch of things to identify a collection of new axon injury signals. And of course, we can't do this without the expertise and the reagents and tools of the Milbrandt Lab and how much they've pioneered primary neuron high content screening. And so if we want to screen in a dish, we need an assay. So how do we do that mouse double nerve injury assay in a dish? And I'll walk you through that here with this fancy cartoon. So we take neurons from a mouse, and these are called dorsal root ganglion neurons. They are the neurons that I described to you at the first slide, where they have cell bodies in your back and then extend into your extremities. And if we take these neurons and we cut them out of the mouse, that in itself injures the axons. And so they're going to activate injury signals, drive the regeneration program. And if we look at them in a dish after 18 hours, they look like this. So here's the cell body. These are the axons extending from it. They don't look very long. It's because at this time point of 18 hours, the regeneration program is not quite on. So it takes some time for all that signaling and all those genes to turn on. But by 24 hours, the program is on. Well, how do we know that? Well, we perform our second testing injury. So in a dish, this takes the form of replating the neurons. And how this works is we have neurons in a dish, like you see on TV, those Petri dishes, although they don't really look like that in actuality. The neurons are ripped off of the plate, their axons are torn off, we put them in a new plate, and then they regrow axons. And now if we look at these neurons after the same 18 hours in culture, they look like this. So the only difference between these two cells is these guys have the regeneration program on at the time of injury, and these guys are still attempting to turn it on. So for our purposes, it's still off. And this gives a nice, somewhat binary readout that we can then quantify by tracing the length of these axons. And so now, how do we turn this into a loss of function assay? Or how do we break something and then see if this program still turns on? Well, we can do that by, for example, adding a drug. So drugs are small molecules that bind to proteins and inhibit them from functioning. So what if we inhibit DLK, which we already know is a known injury signal that drives the regeneration program? We can apply the drug into the cells during this 24-hour period when they're trying to turn on the regeneration program. We wash the drug out during the replating, and now, after 18 hours of culture, they look like this. So these neurons were pre-treated with the inhibitor to DLK, and then grew axons in the absence of any manipulation. And so all we did was inhibit this signaling phase, where the neurons are trying to turn on the regeneration program. And effectively, we turned it off. And this assay was really pioneered by the people shown on the bottom of the screen here, particularly by Aaron, who did a wonderful characterization of this assay and demonstrated that it recapitulates a lot of the key hallmarks that the in vivo or in animal assay does. And so what I wanted to do was miniaturize this assay, so make it really small so we can look at a lot of things at once. And I wanted to look at a whole bunch of drugs and find things that looked just like DLK inhibitor. And so ultimately, the protein targets of those drugs would then be new axon injury signals. And then we can perform follow-up analysis to determine how they work. So how did I do that? To miniaturize this assay, we perform it in these things called 96-well plates, where each of these is an individual condition, and you can have a separate drug, and so essentially you can test 100 things at one time. Here's our culture pipeline, which I showed you on the previous page, but it has some nice little timeline things added to it, where we plate neurons, and then we give them 24 hours to activate their regeneration program, during which we apply a drug, one drug at a time, from a huge collection of drugs that we call a library. Then we wash the drugs out, we assess the status of the regeneration program by injuring them a second time in the form of replating. We let them go 18 hours, and then we fix them, which essentially means we freeze the neurons so then we can take pretty pictures of them. And so that's what this looks like. So here we can use techniques in the lab to make the cells glow certain colors. And you can see neurons shown here in red. Might be a little hard to see it in the back of the room. But all of these are axons. These are the cell bodies, and we can also identify nuclei in this white channel here. Then we take this plate and we put it in this giant box in the Milgrandt lab, which will then automatically image every single well by itself and spit out thousands of pictures. And then I can go relax and then tell Jim that I'm working. And then once we have all those pictures, we feed them into this automated neurite quantification pipeline, which I built in this software called Cell Profiler. And here's the logic of how that works. So this is a raw image. The program will then identify the cell bodies using the red and the white. So everything that's red and white becomes a neuronal cell body, and then all of the red gets traced as neurites. And here are those tracings overlaid with the original image. And then essentially, all of it gets summed together, you divide the values, and then you get a mean neurite length per cell. So you get this value for each one of those individual wells on that 96-well plate. So before I show you the results of the screen, I want to show you what the controls look like, because those are usually required for screening efforts. You want to have something that doesn't work, and you want to have something that you know works. And so what we use as our controls, so here's what the plate layout looks like. So each of these wells would be one compound from a library. So these are things we don't know what the effects will have. But down here are things we know what they should look like. So as a negative control, we have this compound called DMSO. It shouldn't do anything. And as a positive control, we have an inhibitor to this protein called JNK. And JNK is downstream of the DLK pathway. We couldn't use the DLK inhibitor that I showed you earlier because at the time it wasn't commercially available. So we use an inhibitor to junk, which is also a very well characterized axon injury signal that drives axon regeneration. So when we treat cells with either of these compounds, this is what they look like. These neurons treated with DMSO, their regeneration program activates just fine. They grow very long neurons. That's great. However, when we treat with an inhibitor to junk, we strongly attenuate the growth in these neurons. And here's what the data looks like. So each of these dots is a single well across the entire screen that I performed. Everything is normalized to 1. So 1 means it grew well. The program was activated, everything worked as it should. Everything that's lower means it was inhibited. And you can see that we can reproducibly inhibit activation of the regeneration program with junk inhibitor. And so, that time is not right. We have not been here for 45 minutes. Anyway, so this is the same data plotted as a histogram showing that that our positive and negative controls form distinct distributions. And so this really tells us that our assay is able to detect things that have no effect and things that block the program. And so now we're confident that using this assay, we can find things that we want, which would be other things that look like junk inhibitor. So here are the results from the entire screen. I screened a library of 480 compounds at 2 doses. So each of these dots is one of those conditions. Again, everything's normalized to 1, so all these activated the program, so most compounds had no effect, which is good. And then this marker at 0.5 is anything that grew 2-fold or less than the negative control. And I determined that that would be worth following up on. So all the things here in green, we designate as hits, or things that we would like to investigate further. And so here's a quick summary of all the hits that I found. First, I'm going to start with, we found a number of inhibitors that targeted known regeneration molecules, such as MAP kinases, JAK-STAT signaling, and the mTOR pathway. And all of these are very well-studied compounds that are injury signals. And so finding things that you already know about from your screen is very good because it shows that your screen was accurate and it produced what you expected. Now, if that was the only thing we found, that would be every screener's worst nightmare because you didn't find anything new. So fortunately, we did find a number of compounds that targeted unknown or targeted proteins that had no known role in injury signaling. And the top hits in the screen were 2 inhibitors to HSP90. And they are called galvanomycin and a galvanomycin analog. So I don't have to say that a million times throughout the talk. It's going to be abbreviated as 17-AAG. And so in addition to being the top hit in our screen, why did I choose to follow up on HSP90? Well, the HSP stands for heat shock protein 90, and it's a molecular chaperone. But unlike traditional chaperones that help fold new proteins, HSP90 functions later in the protein pathway and specifically helps facilitate the function of signal transduction molecules. So things like kinases and transcription factors. And this made sense to us because if we wanted to find new axon injury signals, well, we found this chaperone that helps facilitate signal transduction. And moreover, there's no known role for HSP90 in axon injury signaling or axon regeneration in general, and actually very little about HSP90 in axon biology at all. And so this was a really novel thing that we thought might add a lot of understanding to the field. So what do we do after we perform the screen? Well, first we have to verify it by doing it in the low throughput assay. So this means not using any of the fancy high content robotic tools and all that stuff. We do it by hand, and you can see here, we treat with DMSO, axons are very long, they turn the program on. When we block with HSP90 inhibitor, we see the same thing. And to confirm that HSP90 inhibition was giving us this phenotype, we purchased a structurally independent HSP90 inhibitor that I'll abbreviate as GT, and it also gives the same phenotype. And here's DLK as a control. And so this demonstrates that we've blocked the functional readout of the regeneration program. But what about all those genes I told you are part of the regeneration program? Well, we can assay those using a similar approach. We can plate neurons and give them 24 hours to turn the regeneration program on, and then we can assess gene expression using this technique called PCR, or we can stain for molecular markers of the regeneration program, which basically means make proteins look pretty colors, and then we see which ones are brighter than the others. So first I'm going to start with some transcript data. So these two genes, you don't really need to know the exact function of them, but they're very well studied regeneration associated genes that are hallmarks of an activated regeneration program. And you can see that they're robustly induced in injured neurons versus uninjured neurons. And importantly, when we inhibit HSP90, we potently block the induction of these genes. And as a control, that also happens when we inhibit DLK. And so next, what about those fluorescent markers? Well, one thing we can look at is the transcription factor Jun. It's downstream of DLK and jump signaling, and it's absolutely required to activate the regeneration program. And so here we can probe for activated Jun, and Jun is activated via phosphorylation. So here we can see neurons in green, and then this red signal is that phosphorylated or activated Jun. In white is just the Jun signal, so it's a little bit clearer to see. You can see uninjured neurons have no induced Jun. 24 hours after injury, there's a lot of it. But then when you inhibit HSP90 or DLK, you don't see any Jun activation following injury. Similarly, we looked at two other molecules. Again, their exact function is not important for this talk, but just know that both of these are robustly induced. You can see how bright this signal is here compared to an uninjured neuron. Same for this one. But interestingly, we see no effect when we block either HSP90 or DLK. So collectively, our data demonstrates that inhibiting HSP90 prevents the regeneration program from activating. But this suggests we might not be shutting down the entire regeneration program because other pathways could work in parallel that are responsible for activating these two proteins. And so next we wanted to start thinking about mechanism. HSP90 is required as a component of injury signaling, but how? And so this data led us to a clue about the possible mechanism in that HSP90 inhibitor and DLK inhibitor show similar selectivity. And what I mean by that is that they block the same things, but have no effect on the same things. And so inhibiting HSP90 really looks a lot like inhibiting DLK. And thinking about the mechanism of HSP90, in that it's a molecular chaperone that facilitates signal transduction molecules, which includes kinases. Well, DLK is a kinase and is required for axon injury signaling. And so I hypothesized maybe HSP90 chaperones DLK, and that's why it's required for axon injury signaling. So how do you demonstrate that something is a client of HSP90? Or in other words, demonstrate that HSP90 chaperones whatever your protein of interest is? Well, there are two main hallmarks of that. One is the two proteins bind. So is there a physical interaction between HSP90 and DLK? And the second is when you lose HSP90 function, the target protein should degrade. And so next I'm going to show you data that addresses these two questions. First, does HSP90 bind DLK? The answer is yes. So this experiment is a co-immunoprecipitation experiment. It essentially means we use an antibody that detects DLK. And so we have neurons, we let them get comfortable, and then we explode the neurons and collect all their protein. And then we take that protein soup and we take an antibody and we remove DLK from that soup of proteins. And when we do that, we obtain DLK, but we also bring along with it HSP90. So that demonstrates that HSP90 and DLK interact with each other. And this happens in neurons at endogenous protein levels. So next, what about, do we inhibit HSP90 function? Can we see a loss of DLK protein? We also see this. So this experiment is performed by plating some neurons, letting them get comfortable, and then we apply some drug and again collect protein from these neurons. And here's what that data looks like. You can see over 8 hours when we treat with the control compound, DMSO, which should not do anything, each of these bands is the amount of DLK in the protein. DLK is stable over 8 hours, and it's quantified here, so the amount of DLK is not changing. However, when we inhibit HSP90, we see a steady decline in total DLK protein. And you can see it drops quite rapidly. And so next I wanted to ask, are we depleting the existing pool of DLK, or are we just blocking synthesis of new DLK? And so to answer this question, we can block total protein synthesis in an alternative way using a drug called cyclohexamide. So this drug will block any new protein from being made. And you can see over the time period of 8 hours, DLK is not turned over within that time. And just to confirm that cyclohexamide is working in our system, we can block for a labile protein, SCG10. That's turned over very quickly. And so we can see that treatment with cyclohexamide depletes SCG10 levels almost immediately. However, DLK levels are maintained. So collectively, this data demonstrates DLK is not turned over within a period of 8 hours. However, when you inhibit HSP90, you can rapidly deplete that existing pool of DLK protein, suggesting that HSP90 is required for DLK stability. And so lastly, I wanted to demonstrate this in vivo. So I injected HSP90 inhibitor into mice, and I collected their sciatic nerves, and again, looked at all of the protein that was in the nerves. And you can see, compared to the controls, when we inhibit HSP90, we see a pretty significant drop of total DLK protein in these nerves. And so together, these data demonstrate that HSP90 chaperones DLK. And this is a possible mechanism by which HSP90 is required for axon injury signaling. Now, the one thing is, all of this data relies on pharmacology. We're using an inhibitor to HSP90. So I wanted to demonstrate, is HSP90 genetically required for these mechanisms? And so, unfortunately, there's 3 HSP90s in mice, which makes genetics a little difficult. But luckily, being in the D'Antonio lab, we have flies to work with. And so Captain Russo performed this experiment for me. So what you're looking at is a Drosophila larvae, which is essentially a slug that will pupate into an adult fruit fly. Sorry, Aaron, but slug works best for them. So anyway, this is its brain, and it extends its axons out. And so we can drive RNAi to HSP90 in the nervous system of these flies. So essentially, we're depleting HSP90 gene product in these animals. And you can see here that HSP90 protein is lost when we do that. And concurrently, we see a loss of DLK protein. So this demonstrates that HSP90 genetically is required for the biochemical phenotype of DLK stability. But what about axon injury signaling? That's how we started on this process to begin with. Well, we can perform a similar assay in flies, again using these larvae. So here's the same picture of this larvae, but now it's flipped on its stomach. Again, here is its brain, and in here there's a lot of motor neurons that are going to extend axons out into the muscles of the larva's wall. So what we can do, or what Alex can do, is take forceps and crush these axons. And just like in mammals, the axon is injured, injury signals are activated, they're transported back to the cell body, and they drive the pro-regenerative program. And we can assay junk signaling, which is a readout of injury signaling in these animals, using this really cool junk reporter, which glows white in this panel. And so what you're looking at here in green is all of these neurons that are located here. And when you don't injure any of the axons, you see no signal in this reporter channel. However, when you crush the axons, 24 hours later, you see a robust induction or a glowing of this junk signaling reporter. And it's overlaid here. So it's yellow here and white in these pictures. So importantly, now what happens when we remove HSP90 in these flies? Well, as we expected, we can see a complete block of junk signaling. And as a positive control, when we block DLK, this should also have the same effect. So collectively, this shows that HSP90 is genetically required for DLK stability and axon injury signaling in mice and Drosophila. And so one thing I haven't told you so far is how important DLK is to a variety of neuronal phenotypes. So far we've only talked about axon regeneration. But as you can see here, you don't have to pay attention to all of these proteins down here. But I just want to use this picture to illustrate that DLK is essential to a number of other mechanisms that occur in neurons. Some of them are also in injury and disease, so they are required for cell death or degeneration. But also DLK has a separate life during neural development. And so what I wanted to know was, have we uncovered a mechanism of DLK regulation that's specific to axon regeneration, or have we uncovered a that's cool, uncovered a novel general mechanism of DLK regulation? Because that would potentially benefit all these fields, because a lot of people are understanding DLK and really want to know how it works. And so this could possibly benefit them. And so essentially I wanted to test this in a non-injury paradigm, which means development. And again, going back to our fly expertise, we have a really convenient developmental, completely DLK dependent phenotype that we can test for. So what we're going to look at here is something called the neuromuscular junction, which essentially is where a motor neuron attaches onto a muscle and then fires to enable muscle contraction. And this is what it looks like in the fly larva. What you're seeing here is a nerve stained in red, and off of that nerve is a single axon that's going to shoot out and synapse onto the muscle. You can't see the muscle because it's not stained in a pretty color, but all of these green dots are essentially synaptic connections, or where that axon is touching the muscle. And a while back, Aaron discovered a really cool mutant in these flies called high-wire flies, where they have a loss of function in this gene called high-wire. So essentially, high-wire is broken in these flies. And as a result, these synapses are majorly overgrown. And you don't have to be a neuroscientist to appreciate that if something's supposed to look like this and it looks like this instead, that's probably not good. And so later, Aaron's lab discovered the mechanism by which this process happens. And it turns out that high-wire negatively regulates DLK, or high-wire targets DLK for degradation. And essentially, when high-wire is broken, you have too much DLK, and then you get this overgrowth phenotype. Well, how do we know it's DLK specifically? Well, if we get rid of DLK, you can completely suppress this phenotype, or essentially, you revert it back to what it normally should look like. So now the question is, if we knock down HSP90, can we see the same effect? And we do. And this is you can quantify this overgrowth by counting the number of green dots here and plotting it over here. And you can see that when we lose HSP90 gene product, we lose total DLK protein. And that's why we can suppress the phenotype and make it look just like loss of DLK itself. And so to summarize, what I've told you today so far is that we've uncovered that HSP90 is a component of axon injury signaling. And I demonstrated a possible mechanism by which HSP90 facilitates axon injury signaling, in that it chaperones the essential axon injury kinase, DLK. This mechanism is evolutionarily conserved, which is a fancy term that we use to say that it happens in multiple organisms across the evolutionary time span. So it's in people, it's in mice, and it's in lower organisms such as flies. And so this mechanism may have existed for quite some time. And then lastly, with the high-wire data, I showed you that DLK can be regulated by HSP90 in non-injury contexts. And so before I wrap up, I just want to revisit the high-content assay we used to arrive at this point. And you can imagine many things that you can do with this assay. So 480 compounds is really small, so you can expand that and screen larger libraries. You can also use different readouts in addition to growth. And you can imagine taking this assay to a variety of new places to uncover new aspects of axon regeneration and the signaling that occurs within it. And I know at least one of you out there is going to ask about this, so I'm going to preempt that question, as I've learned over the last 3 years of giving this talk. What about this guy? Did you find something that boosted regeneration? Well, no. I repeated this and it didn't repeat. And I just want to draw your attention back to the controls and specifically point out that there's a number of outliers, even in the control condition, where they show above average growth. And so it's not uncommon that out of a huge data set, you might get a few that end up being up there that are not really real hits. So they're not having an effect on this program. Alternatively, though, we can adapt this assay into a gain-of-function format. Or essentially, we can use this assay, change it up a little bit, and find drugs that activate the regeneration program. And indeed, that's what Erin and I did. So she found out that when you plate neurons, the program is active by 24 hours. However, if you leave them in culture for about a week, the program then turns off. And so now you have uninjured neurons in a dish, and you can add drugs and then do the replating assay, high content imaging, everything I just showed you. And then you can ask, can we find drugs that induce the regeneration program in the absence of injury? And so I performed that screen with Erin, and she did really nice work following up on the mechanism of our top hit and published a really nice paper recently, so you can talk to me or her, more her if you would like, about what we found and how that assay works. So to summarize the last few years, we sought out to find new axon injury signals. So what are the things that are turned on by injury that activate a pro-regenerative program in peripheral axons? And to do that, we developed a high content assay where we can essentially injure neurons in a dish and find things that are required to turn on the regenerative program. And from that work, we uncovered HSP90 as a component of axon injury signaling, the first implicated in axon regeneration in general. And lastly, through mechanistic analysis, we uncovered a new mechanism of DLK regulation, which could benefit other fields, not just axon regeneration. And that's it. That wasn't so bad, right? We can do it in 10 minutes. Okay, so graduate work is not a one-person thing. I'm up here by myself, but there's an enormous amount of people that made all of this possible. Of course, my lab, but I'll have more things to say about you guys in a few minutes. Milgrandt Lab, they were really instrumental in really bringing high content in vitro culture to our lab. Erin's lab has done a lot of fly genetic screens, but as he said earlier, this was brand new to us. So multiple people, especially Dan, Willie, and Josiah, were really critical to getting this project off the ground. And I especially want to thank Willie and Zach for helping develop the cell profiler pipeline. Willie introduced it to me, and then he left, and I freaked out, but then luckily Zach joined the faculty and saved my ass quite literally. So thank you. Don't give me that face. But anyway, thank you to the two of you. The High Throughput Screening Corps provides drugs to us, so thank you. Of course, funding is a necessary requirement for all science, so the lab's funded by the NIH, and I specifically want to thank the Needlemans for an extremely warm and generous donation to the DevBio program to support students like me. Of course, my thesis committee, thank you to all of you for everything, for contributing all of your unique expertise that really contributed to the well-roundedness and success of my project, and for being positive about my data when I was not a lot of times. So your support was really, really helpful. And thank you to Jim for being a great chair and running the show. DevBio program, Jim and Carrie run an amazing graduate program here. It is the best. You have to look at the data. It doesn't lie. So, and along with Sally and Stacy, all of you make such a tremendous program, and I'm so fortunate to be a part of it. And then lastly, the department itself has been so warm and welcoming. It's been a great opportunity to be in the Department of DevBio. And I don't see any other departments taking everyone to the botanical gardens for Christmas. So, I'll just say that. So thank you to everyone here. And now we have some specific people to thank who contributed just to my personal journey and scientifically, and made me survive grad school. So first up is Sally for being the best DB mom. Of course, beef beef, Festus, and I don't have a picture of Jasmine, I'm sorry. Amos, and of course this crazy guy who lives with Sally. Just comes out of the woodwork sometimes. But anyway, you and Bill have been so warm and generous, and we love going to your house every year, and she's the only one who puts up with Jim's bullying. So everyone thanks Sally after this.
Thanks for that great picture.
This was the mild one. You know the other one that I was respectful enough not to. Okay, so these fools, the D'Antonio Lab has been amazing to be a part of. I've made friends and colleagues for life, and everyone is just so warm and talented. Like it's just the best combination you can have in fellow scientists. EJ wanted to learn cell culture one day randomly, and I thought that was hilarious. And he's never been back since. So I did something to him. Oftentimes, lab is too fun, which inhibits productivity occasionally. But a number of you have been really close friends to me and make lab just such a joy to be in. Shout out to Lab Breakfast. This is a tradition we started. We have a massive breakfast. We make fresh eggs, fresh pancakes. Everyone brings food, and then we have enough food for 18 months. So, lab breakfast forever. And so within the lab, there's two people I especially like to thank for essentially making any of my data possible. I know it's probably not good to say it in front of my committee, but like the two of these extremely talented postdocs were instrumental in nearly every aspect of my project. Aaron and I started about the same time, and now we're finishing about the same time. We've been through screening together, which before I said, oh, it's so exciting, and look at all these fun pictures. The day-to-day sucks ass a lot. So thank you for being just the best partner to go through with that and overcome the toughest days. We literally climbed a mountain together, and we brought our Southwest snacks along with us because that was important. And then Dan is probably the most talented scientist I know, and he will never acknowledge that, but you are. And anything biochemistry that you've seen had some influence of Dan teaching me how to do it right or nudging me away from terrible mistakes. But really, and I bugged him. I don't know how he still has his ears, but I have bugged him about everything and anything incessantly. You're going to have an amazing lab, Dan. And of course, all that genetic data, which is, you know, somewhat important, wouldn't be possible without one special person. Alex has been instrumental in all the Drosophila experiments, and just a really tremendous friend. Some classic beardless pictures here. But thank you, Alex, for everything and serving up that knowledge. Okay, so the D'Antonio Lab wouldn't be what it is without the man himself. This is an OG picture. This is at Vera's defense, where he took an enormous piece of cake and dumped an entire cup of fondue chocolate over it and walked out the door. And said, about to get Father of the Year right here. I don't know how you got it home. But anyway, it's been tremendous working with Aaron. I don't have enough time to explain such an amazing influence he's had on me. He's taught me how to do science, how to think in the big picture. And most of all, he has this weird ability that everybody hates him for to make you feel better about your shitty data. You have a piece of paper and you just, it's the worst. I'm done. I'm gonna go show Aaron so he can say, yeah, that does look a little trashy, but it'll be okay. But you go in and 10 minutes later, you're walking out. Well, I don't really need that group. And that one we could do again, and that's not that bad. But this one was hard, and this is the one that worked. Okay, we'll be fine, we'll be fine. I don't know how you do it. Maybe your guest chair is laced with Xanax or something. But he has the most uncanny ability to help you see what's really important and find the critical experiments. And I will be forever grateful for teaching me how to be a great scientist. So, thank you, Aaron. All right, we're starting to cry, so we're going to speed it up. All right, this guy. Oh man, so I don't even know where to begin with Jim. In a good way. No smirking at me. Jim is the heart and soul of the DB program, and for many people, WashU in general. This man is the most selfless faculty I ever know and probably ever will know. He puts students above himself, above his lab, and above bureaucratic bullshit that happens at any institution. And he does it because he loves students and he mentors out of love, and he's a born teacher. And he's been an amazing mentor and friend. I'm a better writer and speaker. I don't know anyone who's been in contact with Jim who is not. After their time with him, and he takes us fishing, he goes to game night. Oh man, he kicked everyone's ass this time. This was a good memory. We can talk about that later. He takes us for ice cream. We compare rocks, his favorite activity. But Jim will forever be just a lifelong colleague and friend. And thank you so much. All right, so now is the point where people normally think they're friends, but I prepared in advance by being a crotchety old bastard this whole time, and so I didn't make any. So that means we get to proceed directly on to Bruce. So, Bruce, you've been like a son to me. Oh, God damn it. You can't escape this guy. All right, fine. We'll do friends, but quickly. All right, thank you to my friends for keeping me sane. You've given me some of the best memories in grad school, and I can't thank you enough for it. Specifically, Ryan and Tara and Gav. I got a picture of Gav, the white whale. You don't understand how hard this is. Rem, and of course, Neats. Oh man, Neats. And yeah, thank you. And it's just good to have, everybody says this in all their defenses, but you need fellow graduate students to go through some of the unpleasant moments that all of us are going through. Luckily, I got good people who know how to deal with that appropriately. And special thank you to these two, Michael Bahavna and the other half of our 4-person duo. This is at Kieran's wedding. A little late. And a little bit of booze there. And this, yes, we did wear hoodies backwards so we could fill the hood with popcorn while watching a movie. Also, maybe alcohol there too. Okay. So, lastly, one last friend, this guy. So, Blaise and I have been friends since the first day of undergrad, and he's been with me ever since. And then we grew up. He has a real job where he makes money. And I have whatever this is, where I at least get to make fun of him for a minute. But he's visited a few times, and we still talk every single day. He's been such an important part of my life, and thank you. Yeah, look how cute this picture is. Okay, we're almost there, I promise. Of course, you can't do this without family. Everybody's been so loving and supporting. And the best part, nobody asked me when I was going to graduate. So thank you. They all asked, how are things? How is Anshu? How is school? Are you enjoying it? And those are nice questions. Are you paying attention, Mom? But anyway, thank you to all of you, especially Erica, Ronnie and Shelley, Carrie Siddell, and Ina and Rick, and these two princesses. And then speaking of family, the most important parts, and Aaron's two favorite people.
Go back to the
These two guys. So, Bruce and Max, you make even the worst days bearable because look at this. Like, what is this? They're just the most wonderful little bugs you could ever ask for. And of course, back at home, Zoe and Max. But thank you to Max and Bruce. I love you both very much. And two people I am indebted to endlessly are these two, my mom and dad, and I think raised me okay. Some people might have some objections, but yeah, they pursued or influenced me or inspired me to do something intellectual or something with my mind, because I don't really have much else to work with anyway. So, so my mom inspired me to become a scientist, and because of my dad, I tried to do it while making jokes the whole time and having fun with things. So thank you to you both. One last person. Anshu has been the greatest part of grad school. We've gone on adventures together. Thank you for experiencing new things with me. Thank you for almost cooking Bruce, deciding against it at the last minute. Despite your reservations about the two of us, you stuck by me. And you believed in me and my science when I did not a lot. And thank you for making me grow a beard to hide my awkwardly childish face. And then, most importantly, thank you for being yourself and for being possibly even crazier than me. And making me laugh and smile more than I ever thought I could. So, with that, before I lose it, I have kept it together. You have no idea. Making this, I was bawling. But anyway, I will take your questions now.
Questions. Just want to invite everyone back here at noon. We'll have lunch and cake and all sorts of goodies, so please come back.
Aaron's gonna dance. Really? Yes. Yes.
Where do you see the next steps going and in terms of therapeutic value?
That's a fair question. I don't know. So she asked, where are the next steps in therapeutic value? The next steps for mechanism, there's more to learn about how HSP90 facilitates DLK function. And for example, it's required for its stability, but is HSP90 required for DLK's kinase function? So does it just help it stick around, or is HSP90 required for DLK to phosphorylate its downstream targets? Therapeutic value, for this case, I don't think HSP90 is quite the best approach, because there's a lot of clients HSP90 interacts with over half of the human kinome, which is hundreds of things. And so if you wanted to target something like, for example, DLK is a major driver of several neurodegenerative diseases, and you might think, well, can we inhibit HSP90 and then fix those? But you would really want to inhibit DLK itself, because when you treat something, you want to inhibit as little as possible. You want to be as direct as you can with your treatment. And if you use HSP90, it hits a lot of things. But HSP90 is used in the clinic to fight cancer because it's also required to stabilize a ton of cancerous proteins that get made. So it is used for that, but I would bet because it affects other things like DLK, which sometimes is required in a good context, you might not want to target that. But yeah, we can talk more about that later too.
Thank you.
All right, we'll turn it over to
Okay.
the committee.
That is fine. We're over here. All right, we'll do that in a second.
I don't know.
You have a Mac?
Yeah,
Did you just say screw the imaging?
no, I have to go, but
Oh, okay.
we're starting 10 minutes late. I just want to do.
Yes. I'll log you out.